The Corynebacterium glutamicum is a gram-positive, facultatively anaerobic soil bacterium. Under specific growth or treatment conditions, the wild type produces the amino acid L-glutamate. Metabolic engineering expanded the range of products formed by the Corynebacterium glutamicum. These include now all canonical amino acids, various biopolymers, and an extensive range of other chemical commodities. Solely the output of the amino acid L-glutamate amounts to several million tons per year. This turns the Corynebacterium into a highly relevant biotechnological microorganism. Additionally, over the last couple of years, a couple of new tools within systems biology came out that support the process of reconstruction, refinement, and validation. These include, among others, Model Polisher, BOFDAT, CARFME, or MeMode. Therefore, it is highly desirable to obtain a new genome-scale metabolic model of the Corynebacterium glutamicum to understand its physiology and metabolism at a level where the newly tools can be applied. In this study, we present an extended and updated genome-scale metabolic model of the Corynebacterium glutamicum. This model is based upon the metabolic model by Zelle et al. 2015 and uses newly available tools and applies newly defined system biology standards as defined by Carey et al. 2020. In order to create the genome-scale metabolic model, an automated draft reconstruction was created using CARFME by Machado et al. 2018. Intensive automated and manual curation was performed, such as adding differentiated SBO terms or removing orphan metabolites. This draft was then revised using the metabolic model by Zelle et al. As a reference. All newly added compounds were annotated using IDs from the database BIG. Mass and charge imbalances were manually checked and corrected. Energy generating cycles were removed. Using the tool BOFDAT by Lachance et al. 2019, the biomass was tailored specifically towards the Corynebacterium glutamicum. Quality control was done using the platform MeMode, which allows testing for standardization measures of metabolic models. MeMode evaluates the model consistency and annotation levels. Additional validation of the model was done by testing the growth rate on one full medium and two minimal media. We also tested the model's capacities to produce a glutamate and all other canonical amino acids under aerobic and anaerobic growth conditions. Coming to the results. The overall MeMode score amounts to 87%, which implies a very high level of annotation and cross-references of all reactions, metabolites, and genes. It also includes a finely grained collection of reaction SBO terms which allow to improve cross-reference. The amount of mass and charge imbalanced reactions could be reduced from 170 to 19 imbalanced reactions. These represent 1.2% of all reactions. The model has a stoichiometric consistency of 99.7% and contains no energy generating cycles that end metabolites or orphan metabolites. Our model replicated realistic growth rates on all simulated media under both aerobic and anaerobic growth conditions. And it was also produced all canonical amino acids as well as glutamates. Let's first look at the amino acids. When looking at this graphic depicts the ATP requirement depicted against the CO2 production and the in silico amino acid production. This was all determined using flux balance analysis. Glucose served here as the sole carbon source under aerobic growth condition. The availability of glucose was restricted to an uptake rate of 10 millimole per gram dry weight an hour. The growth rate was also fixed in order to allow the organism's viability. Each square that you can see here represents the result of a single FBA with the objective to maximize the corresponding amino acid production. So our model was able to produce these different canonical amino acids. A different other aspect that is also worth mentioning is the enzyme Pyruvate Carboxylase, which has been reported to play an essential role in the L-glutamide production of the Corynebacterium. We were able to show with our model that when knocking out the Pyruvate Carboxylase, this would only lead to a minor decrease of the L-glutamate production. The L-glutamate production decreased from 7.17 to 7.13 millimole per gram dry weight in hour. So it was only a decrease by 0.04 millimole per gram dry weight in hour. As a brief discussion and disclosure statement, I would like to highlight 2 points. First of all, taking organism-specific information and using a high level of annotation, cross-references, and using also recommended standards really helps to create a model of high quality. And as a second and final point, I would like to say that the high-quality genome-scale metabolic model can ease the knowledge gap-filling process and can help to create more accurate phenotype predictions. Thank you for your attention, and thank you for listening to my presentation.